Buenas tardes a todos desde Sydney y bienvenidos a este salón de actos virtual que el Instituto Cervantes pone a vuestra disposición y que acoge hoy a cuantos amigos estén en disposición de descubrir y de explorar nuevos horizontes de la investigación. Retomamos hoy una de las actividades estrellas de los últimos años. Es una actividad en la que la Asociación de Investigadores Españoles de Australia-Pacífico, junto con el Instituto Cervantes de Sydney, han invertido tiempo, esfuerzo conjunto y, sobre todo, han sido capaces de conseguir frutos, grandes logros, en aquellos distintos encuentros que ya se han llevado a cabo a lo largo de este tiempo. Estos encuentros consisten, para aquellos que no los conozcan, en compartir con todo el rigor científico, pero con la voluntad de llegar a un público interesado y no necesariamente experto. Decía que consisten en compartir breves aproximaciones a investigaciones punteras que se realizan en diferentes campos del saber por investigadores españoles. Estas pinceladas son, sin ninguna duda, una apuesta por la difusión del trabajo de investigadores que en cualquiera de las disciplinas del saber contribuyen con su esfuerzo a ampliar todos los horizontes que el ser humano se está planteando. Tenemos hoy a dos mujeres dispuestas a compartir con nosotros no solamente sus conocimientos sobre biología molecular, también su tiempo. Concretamente, ellas nos van a hablar de sus investigaciones relacionadas con la biología molecular. Hablarán de investigaciones acerca de la temida enfermedad de Parkinson y también nos ayudarán a conocer un poco mejor y a entender qué son los virus, esos molestos y terribles protagonistas que han invadido nuestras vidas en los últimos meses. Ellas son las doctoras Adriana Priego y Marian Blanca. Moderará este debate a posteriori, cuando ellas nos hayan presentado sus investigaciones, la también doctora Carmen Salvado Palomeque. Pero antes de pasarle la palabra a Carmen, que es quien va a iniciar realmente esta actividad del día de hoy, quiero agradecer a todos aquellos que en algún momento han trabajado, han apoyado, han promocionado y de algún modo contribuyen de una forma directa a la realización de estas jornadas. Quiero agradecer de forma muy especial a quienes tan generosamente como nuestras invitadas de hoy, pero también a aquellos que han estado con nosotros en otras ocasiones, han compartido su conocimiento. Y también al público que nos estará viendo y que, como siempre, tiene también la generosidad de compartir su tiempo con nosotros y de acompañarnos. Así pues, gracias a todos por estar ahí, gracias a vosotras, Carmen, Marian, también a Adriana, y gracias al público que nos está escuchando. Sin más, Carmen, cuando tú quieras, puedes empezar con las presentaciones.
Hola a todos. Hello, everyone. I am Carmen. Thank you very much, Raquel, for the introduction. So let me see if I can share the screen. Yep. So yes, Raquel said, I am going to be moderating this research bias session. Thank you very much to everyone for joining us. I'm very excited to be here with you. Yeah, well, I am the one of the board members in SRAP, which is the Spanish Researchers Australia Pacific Association. And I also work in Thermo Fisher Scientific as a field application scientist. So who are we? Who are we? So SRAP is a non-profit organization that commenced in 2014. We have around 170 members with representation in most of the large cities in Australia and New Zealand. And we have strong links with 15 universities and research centers across Australia Pacific. The aim of SRAP is to disseminate excellent quality science and facilitate collaboration opportunities to contribute to the cultural enrichment and knowledge advance of both Australian and Spanish, and therefore European societies. The main objectives of SRAP are to help each other and promote networking amongst researchers and industry in the Australia-Pacific region, to help Australian research institutes and universities understanding the Spanish university system, to participate in and influence the Spanish research system via a network of international associations. And in the long term, we would like to be part of changing the Spanish research system. This event has been taking place thanks to the assistance and support of the Instituto Cervantes, the Australian Embassy, and the Fundación Ramón Arefes, and the Fundación Consejo España Australia. Particularly, we are collaborating in this event with the Instituto Cervantes. This association runs different events, like the Australia-Spain Research Forum, and also runs different scientific awards, like the Premio Malaspina. And like today, we present different Research Bites events in Sydney, and today, the first event online. Here I show you the agenda that we are going to follow in this event. So we are going to have two different talks. As Raquel said, we have Dr. Marian Blanca, and both talks are going to have a duration of around 10 to 15 minutes. And after each section, we will have a final session of question and answer section that it will last around 20 minutes. I will ask you please to write your questions on the question and answer section that you will find in a little icon on the bottom of your screen. And also, it will be good for you to know that this event is going to be recorded in case you can miss it. You miss it and you can rewatch it in another time. So without any further delay, I'm going to say welcome to Marian, the first speaker that we have today. And well, thank you very much, Marian, for joining us, because with the time difference, you are in New York now. We really appreciate the effort with the time that you are doing, thanks for your time. So, yeah, so Dr. Marian is a research fellow at the Mount Sinai Hospital of New York City in the United States. She investigates autophagy implications in neurodegeneration processes. In more detail, she characterized the role of the enzyme LARC2 in vivo using transgenic mice models. This molecule is the most significant genetic risk factor identified up to date in Parkinson's disease. Marian started her scientific career with an Ibercaja scholarship to go to the University College of London, where she initiated her studies in LARC2. She did her PhD in Granada, Spain, at the Spanish National Research Council. In her PhD, she identified the molecular mechanisms of LARC2 mutations associated with Parkinson's disease, and she showed that these mutations show preference with the cyclic skeleton. She obtained another scholarship with the Fundación Ramón Arefes, and then she crossed the Atlantic to land in Manhattan at the University of Columbia to expand her studies in cytoskeleton in neurodegeneration. At this time, she focuses her studies in Alzheimer's disease. Now she continues her postdoctoral adventure situated like in Manhattan, situated near Central Park at Dr. Yu Lab, who is a very well-known researcher in the field of cellular autophagy processes. So here she's going to bring us some Parkinson insights with her talk. That is, the name of her talk is Parkinson, a Shaking World. Please, Mariam, proceed.
Thank you so much, Carmen. for that amazing introduction. Let me see if I can share my screen. Not yet. Okay, it's coming. Okay, so here we go. All right. Let me see. Okay. Is it working? Okay. All right. You can see that, right?
Yes.
Okay. So, all right. So as Carmen already said, my name is Marian. I'm a postdoc fellow at Mount Sinai in the city of New York. Thank you, Carmen, for your amazing words. She just likes me so much. But so yeah, so as Carmen said, today I would like to talk to you about basically what have been my main focus, the main focus of my research during most of my scientific career. So I would like to talk to you about Parkinson's disease, which is the most common neurodegenerative movement disorder. It was first described by James Parkinson in 1817 in his essay on the shaking parsley. And there are around like 6 million people suffering from Parkinson's disease worldwide these days. Now, among those, I brought here some celebrities that I'm sure we're all familiar with. So like the Spanish painter, Salvador Dali, or the actor Michael J. Fox, who is actually one of our main funders for our research these days. So PD is a progressive and age-related disorder affecting around 2% of the population above 65, and that incidence rises up to 4% of people who are older than 85. So as the population ages, PD will become an ever-growing economic burden on society. Now, Parkinson's disease is a multi-factor disease, so it has been described as some genetic factors that I will expand on in a little bit that have been shown to clearly cause Parkinson's disease, but also they have been found some epigenetic and environmental risk factors such as pesticide exposure or heavy metal exposure that have been shown to contribute to Parkinson's disease. Now, conversely, it's interesting to know that there are some studies showing some factors that are characterized as protective for Parkinson's disease. So some of them are some specific genetic factors, but I thought it was curious to show you today that there's some studies that show that smoking or drinking coffee is actually beneficial in Parkinson's disease. And it could actually ameliorate the motor symptoms when looking at Parkinson's disease cohorts. The mechanism is still not really understood, but it's really clear that this, for some reason, actually is protective for Parkinson's disease, which is kind of interesting. But as I told you before, the main factor here is age. So then as the population ages, most of the Parkinson's disease patients would be diagnosed at a late onset. So if we now look at the course of Parkinson's disease, it's divided into two big groups. So the prodromal phase is mainly characterized by non-motor symptoms, including hyposmia or loss of smell, which I think we're all very familiar with these days because this is one of the coronavirus symptoms. People also shows hypotension and also sleeping disorders that would lead to mental issues such as anxiety or depression. Now, as the motor symptoms start to take place, this is when we call the clinical phase. And these some of the motor symptoms that we are all very familiar with include tremor, bradykinesia, which is like slow movement, rigidity, postural instability, that would get more and more severe, leading to eating disorders or dyskinesias. And in parallel, I think it's very important to know that the non-motor symptoms continue to progress. So altogether, making Parkinson's disease a very debilitating disease. So if we now look at the histological hallmarks of Parkinson's disease, PD is characterized by a loss of dopaminergic neurons in the substantia nigra pars compacta, which projects out to the striatum, first to the putamen and the caudate nucleus, that's impairing the nigrostriatal pathway. So they have also been identified some protein inclusions mainly containing ubiquitin and synuclein, which are termed as Lewy bodies. And so, right. So interestingly, I think I wanted to show you today some recent evidence of the literature that actually has come out in the last years, where they found that Lewy bodies are not only present in the brain, but they have also been found in the ENS, so in the gut and in the vagus nerve of Parkinson's disease patients, who actually very interestingly suffer from gut dysfunction as well. Now, in terms of genetics, there are also LAC2 mutations that I will talk in a minute. There are also, these mutations have been shown to increase the risk for Crohn's disease, and conversely, mutations in Crohn's disease-related genes such as CAR15 have also been shown to be associated with Parkinson's disease risk. So altogether, pointing towards a complex crosstalk between the brain and the gut, playing a crucial role in neurodegeneration. So if we now look at the molecular level, this is more and more specific now, but I just wanted to show you a very cute video that I found. So basically, dopamine is mainly involved in two main pathways in the brain. So the mesolimbic pathway and the nigrostriatal pathway, which is the one that is impaired in Parkinson's disease. So just wanted to show you this video. Okay, so we can see that dopamine is this like little blue vesicles that would get engulfed into big vesicles, and then they will merge into the presynaptic neuron to be released into the synaptic cleft, and they would then bind to the dopaminergic receptors at the postsynaptic neuron. Now, this would induce a downstream pathway that would actually lead to the control of movement. Then when this signal gets turned off, dopamine gets traveled back all the way to the presynaptic neuron to be recycled. So Parkinson's, as we all know, currently there's no current cure for Parkinson's disease. And the treatment, apart from symptomatic, involves the use of the brain stimulation, which basically means like stimulating specific areas of the brain to control the motor symptoms, and also dopaminergic medications, including levodopa, which is a precursor of dopamine. So that's trying to restore the dopamine levels in the brain. However, upon long-term administration, these treatments have been shown to induce very chronic side effects, so then highlighting the need of better therapeutic approaches. In terms of diagnosis, Parkinson's disease is diagnosed also at the level of the symptoms, but this is very important to keep in mind because, as I showed you before, basically most PD patients would be diagnosed at the clinical phase. And this is when the motor symptoms are already present. Now, there are more and more studies that show that at a very low level, there's around 30% of dopaminergic neuronal loss at this time. So obviously, one could think how this is already problematic. And basically, what we're missing in the field is better strategies to get an early diagnosis. So in order to shed light to the mechanism, Parkinson's disease have been, as I showed you before, have been characterized by different factors, but I'm gonna focus on the genetic factors. So today, as Carmen already said, I would like to talk to you about LAC2, which is the most common cause of familial Parkinson's disease. Not only that, but mutations and variations in LAC2 are associated with sporadic Parkinson's disease. And importantly, the progression of the LAC2 familial Parkinson's disease is very similar to the sporadic cases. So in some ethnic populations, such as the Ashkenazi Jewish or the Berberic from North Africa, more than 40% of the cases can be due to mutations in LAC2, and variations in the gene further increase risk for sporadic PD. So basically making LAC2 a fantastic candidate for trying to understand the mechanism for Parkinson's disease. So LAC2 is encoded by the PARK8 gene. It was first cloned by a Spanish lady who's Coro Paisan-Ruiz in her Neuron paper in 2004. And autosomal dominant mutations are known to cause late onset PD, which, as I said before, but it's important to keep in mind, it's very similar to the sporadic cases. So this is Coro, who's actually a current member at Mount Sinai these days. And so she cloned LAC2 in her studies in the Basque Country, because actually there's a very high incidence of a specific LAC2 mutation in the Basque population. So this is, I don't want to get too technical, so this is just to show you the structure. LAC2 has two main catalytic activities, GTPase activity at the raw core and kinase activity. Just to mention that all the pathogenic mutations that have been shown to cause Parkinson's disease cluster at this catalytic area, so then suggesting that enzymatic changes could lead to the pathogenic form of the protein. So in terms of the cell function of LAC2, there are many, many studies, but I think kind of like the consensus these days, even though the actual role hasn't really been elucidated yet, it's that LAC2 plays a role in vesicular trafficking through its interaction with specific subsets of RAP proteins. So indeed, in 2016, there were these two RAP proteins were identified to be substrates for LAC2. This was a breakthrough in the field because for the first time they found that all pathogenic mutations consistently increase kinase activity in vivo. Now, this really pointed towards the pharmacological LAC2 kinase inhibition as a promising therapeutic treatment. And this is where all the money from the big pharma is getting invested these days, trying to find a suitable compound. So this is just to show you some of the examples of the beneficial effects that have been already described when using animal models with LAC2 kinase inhibitors. So for instance, in rodent models, LAC2 transgenic mice, kinase inhibitors have been shown to rescue the neural shortening in primary neuronal cultures. Additionally, it has been shown to enhance dopamine release and synaptic vesicle mobility. And very importantly, LAC2 kinase inhibition had managed to successfully reduce the dopaminergic neuronal loss. Now, this is true not only for the rodents, but also for fly models and worms. So in order to screen for a good LAC2 kinase inhibitor, you need to have good available biomarkers. So in the field of LAC2, these biomarkers are based on phosphorylation levels that could be detected by looking at blood samples, urinary exosomes, or CSF from the brain samples. And I just wanted to show you some of my data from my PhD, from my PhD lab. So basically what I found, as Carmen kind of said in the beginning, is that LAC2 display a normal localization, forming these microtubular structures that were actually consistently increased when using a different subset of LAC2 kinase inhibitors. Now, we also found that there was a deficit in cohesion when looking at the centrosomes. And this was true when looking at lymphoblast samples of Parkinson's disease patients. And interestingly, this was reduced when using LAC2 kinase inhibitors as well. So we kind of proposed these two tools as diagnostic tools and that could also be used for screening LAC2 kinase inhibitors. I would just like to leave you with exciting news. So basically, recently they have been found that this company from the Netherlands called Dengue Therapeutics, they have developed two good kinase inhibitors that show good results in terms of safe pharmacokinetics and pharmacodynamics. And so they are actually in clinical trials. Hopefully they will make it to the patients soon. And this is just my take-home message. I hope today I have convinced you that LAC2 is the promising therapeutic target for both familial and sporadic PD, and that the use of kinase inhibitor could be a good, powerful therapeutic drug. And I also thought, and the benefit of time, I didn't really talk about this, but most of the Parkinson's disease-related genes have actually found to play a role in the vesicular trafficking pathway. So as an alternative, therapies targeting this pathway could also be beneficial. And this is just to have a bit of an arty note. So I just wanted to show you this point that I found when preparing my talk. So this illustrated an art exhibition in País Vasco from this guy, and it's called Dardara, which surprisingly is the same name as LAC2 because basically means shaking, like it means temblor in Basque. So I just felt it really nicely illustrated the situation that we are all living at right now with this like shaking times. So I just gonna go with his message. He just said, this is just a temblor, solo un temblor. And I just think that what I really want to leave you with, the take-home message of my talk, is that I hope we have all understood at this time that what could really stop the shakiness is science, and that without science, there's no future. So I just want to thank the organizers for inviting me. This has been an amazing opportunity. Thank you so much, Miriam, Raquel, and Carmen for putting together this event. Thank you to my current lab, Mount Sinai. Thank you for my funders, Fundación Ramón Areces, my previous lab, Columbia University, CSIC, and thank you so much for your attention. And I'm happy to take questions whenever Carmen says that.
Well, thank you. Thank you very much, Marian. It's always a pleasure to hear you. And thank you very much for taking the time. Yeah, we will have the round of, it's a very interesting research. For sure, it's very crucial to try to help people. And but I agree, without science, there is no future, right? Nowadays, we truly realized more than ever. So, yeah, we will have the opportunity of asking Marian questions after Adriana's talk, and in this last session of 20 minutes. And please, I'm going to ask you to write the questions that you have in the Q&A icon, and I will read the questions for a better, to facilitate and to organize better the event, so not everyone speaks at once. And yeah, well, now I'm going to introduce our second speaker. Hello, Adriana Priego, Dr. Adriana Priego, how are you?
Hello, good afternoon, good morning, with whatever your time zone in the world.
Yeah, thanks, Adriana, for joining us and give some insight in this hot topic that we have today. Everyone hears about viruses, and yeah, it's going to be a great opportunity for us to learn a little bit more, and yeah, perhaps asking some questions at the end of your talk as well. So I'm going to introduce Dr. Adriana. She works as a research fellow at the University of Queensland in Australia. Since 2019, she has been working on her favorite topic, viruses. So her research is based on studying antiviral factors on the human immune response against dengue virus. She studies which scenarios of this immune response lead to patient recovery or mortality in severe cases of dengue. Adriana studied biology at the Americas University in Puebla, Mexico. She did her master's thesis at the Research Center and Advanced Studies of the National Polytechnic Institute at the Mexico DF, on hepatitis C virus infection. And after working in the industry for a year and a half, she obtained a PhD scholarship at the Griffith University in Australia. She moved to the other side of the world to study antiviral agents against transmitted virus through mosquito bites. Her past research interests have been very diverse, including nanoparticles evaluation as antiviral molecules drivers against respiratory diseases, and the study of cancer stem cells that generate leukemia. So Adriana's talk is viruses. What are they? So please, Adriana, proceed with your talk.
Hello, Catherine. Thank you very much for that. Such a wonderful introduction. Yeah, it's happening that I'm on the other side of the world where I was 10 years ago, but here we are and having a good time. Yes, so in a minute, I'll be sharing some.
Takes a little while.
With you, yep. So please, by the end of the talk, all questions are welcome, especially given such times. Please don't know such things as a bad or stupid question. Now it's a good time to ask questions, and I'll do my best to try to answer it in the clearest way I can possibly can. Yeah, I'm waiting to have permission to share the screen. Yeah, so one thing that I would like to people know in advance is that what I'm going to present today, it's about viruses in general. So all the diagrams that you will see are just for the purpose of bringing virus to everyone's attention. If each specific virus causes specific diseases. So again, this is just a very general description of what happens.
Adriana, sorry to interrupt you, but
I know I'm not sharing the screen. I'm just waiting for to have
to get access to the sharing.
I think it was given to
that happened to me as well.
Yeah, okay. Here we go.
Yes?
Yes. All right.
It happens to me before and I got a little bit distracted.
Okay, great. I think we can start now. So as I said, again, this is just, it doesn't characterize as all viruses. So each individual viruses will have its own specific details. So everyone, welcome again. Thanks for your attention and time. My name is Adriana, and as Carmen mentioned, I work at University of Queensland, and I work with viruses. And this plaque, which has 6 wells that you can see here, which has a beautiful purple color, is the closest thing that you could ever be able to see a virus with your naked eye. So obviously it's not a virus, but what you're looking at is the way and the technique we use to quantify viruses. In purple, you're looking at live cells, and each circle or plaque, as we call it, in white, it's equivalent to a virus particle that was able to kill a cell. So that way we count viruses and we're able to know how many there are in our. So a quick introduction of what I would like to share with you today. So we'll talk about what are the viruses. I'll specifically talk about envelope viruses because there are differences between viruses. We'll touch on the structure, how they replicate inside the cell, and with that knowledge, I think I'll be able to show how antivirals work, or at least we're aiming to design these antivirals. And lastly, we'll talk quickly about flaviviruses, which is a family of viruses. Dengue is one of them. And we'll talk about the disease, and I'll share what I believe is a very interesting phenomenon, which is called antibody-dependent enhancement. And we'll talk about what are the implications of this antibody-dependent enhancement phenomenon in the development of vaccines against viruses. So the big question, what is a virus? So in very simple terms, it's a package that contains information. So that's simple. It can have different shapes. It doesn't have to be always a sphere. It can have icosahedral shapes. It can be cylinder, and it can range from a few nanometers to 100 few nanometers. So they're quite variable. They're quite diverse. And not all viruses actually are pathogenic. They are famous because they make us sick. But there's a huge biodiversity of viruses that are not pathogenic to animals. And another big question that arises when we talk about viruses is are they alive? So scientists will talk about two different ways of describing the virus in terms of if it's alive or not. One side will say that yes, they are alive because they have a genetic code that they can pass to, and because they also, viruses are subject to evolution. So evolution, it's pretty much is that force that makes all living things to make it better to survive. However, on the other hand, there's the other side of scientists that argue that no, they're not considered to be alive because they don't have a metabolism. So what that means is that if we were able to isolate a single viral particle in a room, in the vacuum, let's say, well, there's nothing else, not even air, then that particle itself, it's completely absent of life. It can't do anything. It's completely, numb. But that means that viruses actually are required and they need, by definition, the existence of a cell that they can infect in order for them to exist. So this is the structure of an envelope virus that I will focus in today. They're quite simple, as you can see. They don't have many parts. In the middle, in the center, is the information that I was talking about, that it can be either DNA or RNA. And then this genetic information is usually covered by a viral protein called capsid or nucleocapsid that protects this material, this genetic material. And then an extra layer in blue, you can see that it's a lipid membrane. And this lipid membrane actually comes from the cell that it previously infected and from where this virus came from. And in yellow, you can see that they have an envelope protein, which is a viral envelope protein. And some examples of the envelope viruses, the most famous ones are hepatitis C virus, dengue virus, coronavirus, influenza, and varicella zoster. So I wanted to explain quickly what happens inside of the cell when you get infected by a virus. So if you remember, I mentioned that viruses, they completely rely on the existence of the cell. And in order to infect them, they use their envelope viral protein. And this envelope viral protein, it's able to recognize a cellular receptor, as you can see in step number 1. When this viral protein recognizes the receptor, it will then be internalized via an endosome, as you can see here in number 2. And when I say an endosome, in simple words, it's just a bubble made by the lipid viral layer of your cell, doesn't have to be yours. So this virus is already contained in this bubble-like structure, but it's not capable of doing anything. It needs its genetic material to be released in order to make more copies. And this is a very interesting way how viruses do this, and they do so by fusing the virus viral membrane with the endosome membrane. And when they fuse, the genetic material is released, as you can see in step number 3. This fusion of the endosome with the viral particle requires the acidification of this endosome. So if you remember, you have acidic and basic conditions. So when inside of this endosome with the virus, there's a drop in the pH, so it's more acidic, it then triggers this fusion between the virus and the endosome, and then the genetic material is released. This genetic material is then used for 2 very important steps. One is that this genetic material, because it is information, it will be used to produce a protein. And this protein is going to be produced by the ribosomes. The ribosomes are cellular apparatus within your cell, and these ribosomes are they are the ones that are due to make all your viral structure blocks of proteins. So the cellular ribosomes will make viral proteins using the information that comes from the genetic material. Also, this same genetic material, the virus needs to make more copies, exact same copies of it, so that they can then be packaged, and then you have a new virus. So the genetic material will also be replicated into many copies, exact same identical copies, so that this genetic material with the viral proteins can be then packaged, as you can see here in step number 5. And this packaging, then once it's all the viral proteins that conform the package itself with the viral genetic material inside, we'll have some few modifications through this Golgi apparatus. There's no real point in going into detail into those. And then in order for this viral particle to be released, so to be released outside of the cell, it's going to do it via an exosome. And this exosome, very similar to what happens with an endosome. It's like a bubble-like structure that will fuse with the membrane of the cell and then release the virus outside. Okay, so I hope that it was a bit clear what's happening inside of the cell. And while the virus is pretty much hijacking, it's taking over the cellular mechanisms of all the resources and energy. The virus is taking over to make more copies of itself. So when the cell can no longer do its own job to keep herself healthy, then your tissues start to damage. And if your tissues damage, then your organ starts to fail. And that's how we get sick, in pretty simple words. So how do we design antivirals? So the aim of an antiviral is to stop the infection. So the very first thing that comes to mind, and it's the thing that we always try to design, is neutralizing antibodies, because they will bind to step number 1. So that means that these antibodies will recognize the virus, they will cover it, and they will prevent the virus to recognize the cellular receptor. Therefore, you stop the infection. Another option is the example of, you might have heard of hydroxychloroquine. And there are several ways in which this molecule can work. In vitro, so that is in cells where there is no tissue involved, no organ, obviously far less there is an organism involved, just in cells that we can culture in the lab. Hydroxychloroquine, it's been shown that it can actually prevent this acidification of the endosome in step 3. By doing so, it prevents the fusion of the virus with the endosome, so the genetic material cannot be released. Then you stop the infection. And another hypothesis is that because it can also buffer the pH inside the cell, not only inside the endosome, it can then prevent the activity of a viral enzyme that it's responsible of making more copies. So it will act on step 4B. And a third one is the cell-mediated effect, that so that means it's not targeting a specific step of the viral replication, but rather is inducing cell death. So that's a pretty conservative response. So it's like a cell is infected, we're not going to deal with it, just kill the cell and then we stop the infection. And lastly is the anti-replicases, which they prevent the generation of new viral gene copies. And again, these molecules will interact with the viral protein that is responsible of making copies of the genetic material. So it would act on step 4B. And very quickly, this is an image that I wanted to show you. This is what I did in my PhD studies, where I studied the antiviral properties of a plant, extract plant, which is called Melaleuca alternifolia concentrate. For easy purposeness, I'll call it MAC. And as you can see in this image, in blue, you're looking at the DNA. And because the DNA in our cells is compacted in the nucleus, each blue dot is the nucleus of a single cell. And in green, I tagged, so I painted a viral protein. So more green means more virus, more infection. And as you can see here, 24 hours post-infection, there's no real difference between those cells that were infected but with no treatment compared with those that were infected with the treatment. So one day after infection, there's no difference. But there was a significant difference between those cells that were left untreated with those cells that we treated at 2 days and 3 days post-infection. And after several studies, we came to the conclusion that this extract has 2 possible mechanisms. One, it's a virucidal activity, which that means is that it can disrupt the virus before it enters the cell. So that's quite good because you, again, stop the infection. And another one possible mechanism is that it prevents the production of the viral proteins here in step 4A. We don't know how, but we were able to narrow down from all the steps that I showed you, that we believe that this extract is working in this step. Now, again, this was in vitro, so more studies are required to prove this in an in vivo system. Quickly, flaviviruses. So this is a family of viruses, and who are these viruses? Flaviviruses are those mosquito-borne viruses. So you get them when you get bitten by a mosquito that carries this virus. The most popular ones is dengue, Zika, Japanese encephalitis, West Nile, and yellow fever. So in this map, I'm showing you in red all the places in the world that dengue is transmitted. And this is in, well, it's not that current, it's 2015. Due to climate change, this is what it's estimated to happen in 2080. Maybe you don't see much of the spread, but it's getting much, much redder. So there's going to be a huge increase in the incidence of dengue. But there are places that actually be surprising that we could potentially see dengue where currently there is no. One such example is New Zealand, where it's quite south and quite cold. But given to climate change, there will be actually conditions where these mosquitoes can happily live and therefore transmit dengue virus. So dengue infection, there are 4 types of dengue. They're all very similar, but they are just slightly different. That actually we can categorize them as 4 distinct serotypes of viruses. They can cause a range of disease, from symptomatic, no symptoms at all, very mild, like a flu-like, you have fever, or it can actually be quite severe. It can be hemorrhagic or even cause a shock syndrome. And these 2 forms of severe dengue are mainly due to a secondary infection with a different serotype. And this infection with a different serotype causes an antibody-dependent enhancement, which for easy purposes, I'll call it ADE. And of course, this is a problem because the severe disease can actually become deadly. Sadly, it's mainly among children in poor countries, and there's no specific treatment for dengue infection. So what is the antibody-dependent enhancement, ADE? So this is what happens. If you get infected with a particular serotype of dengue, let it be 1, 2, 3, or 4, your antibody will mount an immune response and create antibodies specifically for that serotype. So if you get infected with the serotype dengue 1, your body will create antibodies for dengue 1. And these antibodies will have high affinity. And in other words, it means that they will bind quite strongly and they will be able to neutralize the virus to then stop the infection. Now, what happens, let's say, if I was infected with dengue 1 10 years ago, but yesterday a mosquito bit me with dengue virus 2. So my body thinks that, well, I still have circulating antibodies because I retain immunity from the first infection that are for dengue 1. These antibodies will actually bind and recognize dengue 2, but they have low affinity. And again, in other words, that means that they bind, but much with less strongly, much weaker interactions. And these antibodies, even though they will attach to the virus, they will not neutralize it. And antibodies, when you have this kind of scenario, it can actually be worse than doing good. And this is what happens. So if you have strong affinities, antibodies that recognize very nicely, so antibodies for 1 against virus 1, these antibodies will cover the virus and neutralizing. In the graph, you see 100% neutralization when you have antibodies of 1 against 1. But what happens when you have this mix between previous infection and a new infection with a different dengue serotype? Some antibodies will bind, but they're not able to neutralize. And what happens is that in some white cells of your immune cells, they carry this receptor that recognizes a part of an antibody, of any antibody. So this receptor, it's not the same receptor that we talk about in the early replication, but they actually recognize this part of the antibody. We call it the tail of the antibody. So in instances where you have a virus that it's semi-covered, it's not neutralized with these antibodies, what's happening is that this immune cell is actually swelling like a Trojan horse. It's not realizing that it's actually being infected. So in a patient, you will have your normal infection going on, plus on top of that, this will be happening, this ADE. And this is very important when we try to design a vaccine. So if you just quickly pay attention, dengue dengue has been around for many decades, and we've been trying for that many decades to design a vaccine. Yet, we have not been successful to do so. And why is that? Because this vaccine against dengue needs to be efficient, independently of the state of the patient. So that means the person has been infected by few types of dengue, or has never been infected by dengue, or maybe has only been infected by a few serotypes. You should also take into account the age of the person. So it's not the same if we're talking about a neonate, a newborn, against an elderly person. And also this vaccine must be efficient to give you immunity against the 4 serotypes of dengue, but without generating the ADE effect. So this is just an example to show you of all the safety steps that vaccines actually go through to make it to a person. And as I said, dengue has been around for many decades, but yet none of the vaccines that we've been trying have been able to pass all these safety requirements. And of course, this ADE scenario is shared with other viral infections. So it's not only a case of dengue, but dengue is the one that's most famous and more complicated because you have 4 different serotypes. And so with that, I would like to thank everyone that has come across this crazy path of science that I love. And thank you, Carmen, for the invitation. Thank you for the Instituto Cervantes. I really appreciate the invitation, and all of you for listening. And I'll be happy to take any questions from there. Thank you.
Thank you very much, Adriana. Thank you for your talk, and thank you very much for the contribution on your field. I feel that it's pretty crucial to investigate nowadays. That's for sure. Well, now we are going to proceed with the question and answer section. I'm going to read the questions that you've been sending. And please, if you have further questions, you can just type the name at the beginning of the name of the speaker that you want to ask that particular question. So let me see. Here I have one for Marianne. How many different types of Parkinson exist?
Okay. So I think Parkinson's disease is like, basically there are like 2 main types in terms of what they call the onset. So mostly it's late onset, as I explained to you before, age being the main factor, but there are also some genetic factors that, such as PINK1, PARKIN, they're like specific genes that are linked to the early onset of the disease. So that is kind of in terms of genetics. Now, also, if you happen to have a late onset mutation, but unfortunately are exposed somehow to, you know, some kind of environmental risk factors, that could also make the onset of the disease to actually happen earlier.
Mm-hmm. Thank you. I think we have here Adriana. Do you think viruses are alive? I think she mentioned a little bit at the beginning.
Yes. Well, it's a very philosophical question, to be honest, because they carry characteristics of both being alive and not being alive. I would say that, yeah, I think they are, because in my very personal opinion, if it's subject to evolution, then it is. A rock is not subject to evolution, then therefore it's completely dead. So I think maybe if we are very strict, then the definition of alive needs to be revisited. Yeah.
Yeah. Thank you very much. I have another one here. Like this one is for directed to Mariam. Could you please describe briefly the pharmacological treatment on LAC2 gene for PD, for Parkinson's disease?
Right. So, I mean, I'm not an expert on pharmacologics, but I think kind of briefly, I can tell you that basically the way it works is there are 2 main types of pharmacologic kinase inhibitors. but mostly the ones that have been pushed these days are the type 2, which are basically they're gonna impede the binding. They're gonna compete with the ATP binding pocket, basically. So like inhibiting the kinase domain. Now, I think one of the caveat with the kinase inhibitors these days with LAC2 is that it's just such a big protein that actually the 3D structure, the crystal structure, hasn't been done yet. So like, you know, the field is getting there, and we have like around, it really was discovered like 15 years ago. We already have like clinical trials kinase inhibitors, but one of the main issues is that, you know, the crystallography is missing. So that would really makes it like it would just make it better in the future.
I believe if that's the case, do these inhibitors, do you know if they have off targets? So you've been talking about this, some inhibitors.
Right. So yeah, I mean, right. I mean, yeah, obviously since they started, you know, they have really become more and more specific. Like the IC50 has been low. So yeah, they're getting more and more specific, but yes, there's still some, I wouldn't say off target, because basically what we are working with these days are like very specific. So they actually have what they call on target effects, because basically LAC2 expression, I think I show you on one of my slides. So the expression of LAC2 among the different tissues is very high expressed in kidney and lung. So it's actually what they're seeing in like you know, rodent models and also primates, is that they develop like kidney phenotype. They have some kind of like darkening accumulations, and the same is true for like some accumulation at the lungs, at the alveolar cells. But you know, these days with the new inhibitors, they actually really ruling out these problems.
Okay, thank you very much. Well, with off target is we mean in a specificities, right? So it's they are not that, they don't bind that in such a specific way.
Right.
For those people that probably don't know. I have another question for Adriana. Adriana, do you think that COVID-19 will be like the flu, requiring vaccinations every year?
So again, this type of question, it's really hard to answer, because we have to keep in mind that we're dealing with a completely new virus. So this is the very first time like we've been actually exposed to this virus. So in reality, what we know is very little. Like just recently, there was a publication about HIV, and it took us like 40, 50 years of research to be able to track it down, where it came from, how it does everything that it does, and how to at least stop it. So to answer this question with only being around for 5-6 months on the planet, my answer will be very subjective, and my answer will be that it is possible that it can happen, but not exactly the same. Why? Because with influenza, the vaccine that currently that you have for influenza carries a lot of strains, or again serotypes. Not serotypes, this is actually great, doesn't matter the definition, but it's you know, so they actually, it's not that you have only one, it's not the same virus that you have each year. It's a mix of different types of influenza A virus or B, right? So with coronavirus, with COVID-19, so SARS-CoV-2, I don't think it will be that you need to refresh because there are different strains circulating. It can be a case that you might require boosts because the immunity for coronaviruses, not only COVID-19, so for the family of coronaviruses, it's been shown that for a reason that I don't understand, is that your immunity can only last for 6 to 12 months. So that means, so actually that makes sense. If you think about it, coronaviruses before SARS-CoV-2 were around, and it will cause your normal flu-like, yeah, colds. And you would think that if you would hold immunity for life, at the age of 50, you should never get a cold ever again. But that's not the case. And that's because the immunity for these coronaviruses drops. So I think we might require boosts just to keep the immunity, always effective.
Another question for Adriana. This is a little bit tricky because we've seen that nowadays lots of people talking about coronavirus, the efforts on researching this virus and finding a vaccine and are being huge, and the level of collaboration among scientists is being amazing. No, we've never seen anything like that. That's pretty remarkable and good to see. So in saying that the efforts are like pretty much way bigger than like before, like before having any pandemic event, like any type of the type of investment that we will normally do on a regular disease, right? Without the pandemic. How long till you estimate we could find, or scientists can find a vaccine for this type of virus?
Again,
saying that.
So if I could, I would repeat the same introduction that I gave to the last question. So the short answer will be that it will take at least a year to a year and a half to find a candidate. Okay, so currently, for instance, there are already candidates that are going into clinical trials. So clinical trials, you have clinical 1, 2, 3, and that means that first you need to check that it's not toxic or doing any harm. And so first step is like, okay, it's not causing you sickness. Good. Secondly, then you have to go through a phase where you can actually see a difference of those that have no vaccine against those that have it. So you can't really expose people to an infection. It's ethical, and no. So we don't do that. What we do is the vaccine gets tested, and then we take a blood test, and then we check if your immunity, the antibodies that you produced against that, actually can inhibit the virus in the lab. So if it does, good. Then after that, it has to be done blindly in a massive scale, so in many, many people. So just to do those 3 tests, it takes a lot of time. And that's the main issue here. It's not that we don't have the capacity to come up with ideas on how to create a vaccine, is that we need to go yes to all this process and make sure that it's safe. And then let's keep in mind, okay, one of those, it doesn't have to be one. Let's keep that in mind. There's several techniques that vaccines can be done for viruses. Let's say that from the mainly 4 techniques that are mainly used, 2 of them are actually managed to pass through clinical trials. Let's just hypothesize that. Then we need to be able to produce enough doses to vaccinate a whole lot of a population, from which last I knew was we were past nearly the 8 billion population. So that is a huge amount of effort just to create enough doses. So if everything goes well and smoothly, which it rarely does in science, it will take at least a year to a year and a half. Yes, then it will be a debate of who gets it first. Should elderly people plus doctors and nurses? Right. What about people that already have it? You know, there's a lot of things that we're learning on the way. Right. If we have a safe and effective vaccine, I wouldn't expect it before to a year and a half. Next year, 2021.
Okay, well, thank you very much.
But
I think.
Can I say something, Carmen?
Sorry? What did you say, Marian?
Can I say something to Adriana?
Of course, absolutely.
I was just wondering, like, if this is true, right? So do you think that is there any way they're planning to rush up the whole process due to, you know, the pandemic kind of urgency that we're all like kind of freaking out? Is that a thing, or is that like I don't really know how clinical trials work in the virus field, but do you think that's kind of like a real thing that could happen, or otherwise, I mean, they're just like really selling like something that's never gonna, it's completely unreal, right?
Yeah, so if I get it, you're asking me if it can actually happen in a year and a half?
No, I think she's asking,
I mean, like, can they rush up the process?
Can they rush up the process? So the process is already rushed when we talk about a year and a half. It usually takes 4 to 10 years to create a vaccine.
Right.
So when we're speaking about a year and a half,
Okay.
we can just rush and as quick as possible without cutting edges. That's the other thing. Like, we can't, you know, it's something that people,
Right.
we never cut edges. So that's the most realistic scenario. And again, that is if everything goes
Right.
according to the plan, which again, it rarely does so. But, you know, I mean, like Carmen mentioned before, it's at the moment, it's not like 1 or 2 labs working on it. There's like, I don't know, I lost count of how many labs are actually doing it in every single country. So chances are getting, just by chance, by statistics, it's getting better.
As far as I've seen, they were overlapping the phases of the different clinical trial stages, right? You have 4 different ones, and then they were like overlapping them. So they are like rushing that as much as it can be rushed, right?
Right.
Enough
So
to be safe.
On the pipeline, what will happen is that you'll have clinical trial 1, 2, 3 produce, right? So one way they're trying to accelerate this is you run 1, you wait, see the first, your trial, your preliminary results. Okay, it's safe. We start trial 2, trial 3. Now, if it's looking good, what they've been doing is, it's pretty much like throwing a coin. Like, we're going to start producing doses with the hope that all those 3 steps go well, so that if they get approved, like tick, 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 we have them, the doses ready. But they will never be distributed until those 3 phases are, and that's clinical trial. So that's talking about testing human. There's a whole lot of tests before that are done in ferrets, cells. So it's a huge effort for scientists that are actually working on that. It's a 24/7 job, which, I mean, we always work extra hours, but these guys, they're doing an extra effort.
Well, hopefully the vaccine will be developed soon and this situation can improve. We need science, that's for sure. Without science, there is no future.
We're trying, like, we know if we can borrow, if we can get, so, you know, we're trying to target the address the issue from several angles, not just not just a vaccine.
Yes, there are. The entire world is trying different vaccines and different methods. So just quickly, just to wrap up, I want to ask you to, like, if you want to tell me quickly, what's the biggest challenge that you faced as a scientist, and what's the most, like, the part that you enjoy the most? What's your favorite aspect of being a researcher and doing science? So very briefly, like in a couple of lines, what's the best and what's the biggest challenge?
Marian goes first.
Please, Marian.
Go first, Adriana. I think that's the hardest question.
To close with, like, please, Marian.
I mean, that's a hard question. I don't know. I mean, Carmen and I, we actually talk about it a lot. So, I don't know. I would say, you know, as Adriana said, we all put like extra hours. We work around the clock, and we still like really enjoyed it. For some reason, it pays back. So yeah, I mean, that's what I enjoy about science. It's just science. I think there's a kind of, it seems like almost like a romantic relationship in a way. And I think what I don't enjoy, and I really, really hope,
the biggest challenge
changes, is the system. I think the system is just, it's bad. Just as that. So I mean, it's not okay that because we love it, we just have to be taken for granted, like paid not as we should be, etc., etc. So, you know, the system is complicated to actually get to the top. There are really brilliant people who get out, as you know, like many people that we know. So I think we really need to work to change the system.
Absolutely. Thank you very much for your response. We understand. Adriana, please, can you tell us what's the thing that you enjoy the most doing your job as a scientist? And what's the biggest challenge you face?
Yeah, the biggest challenge I face, I think it's I don't want to copy Marianne, but it's within those lines that yes, we deal with working contracts that are like horrible. Like we don't have a regular normal long life contract. If we have good luck, we have a 3-year contract, and that was being lucky. A lot of scientists, they have to live with a 1-year contract, and we are expected to produce so much data and help, cure diseases, but our work safety is just not okay. So yes, I think that's something that you have to be aware when you become a scientist, that sadly that's the reality, that you need to kind of have always a plan B, C, and D, because you do your best. It doesn't mean that you're a bad scientist, but you might run out of a grant, you didn't get a grant, and then that's it. But of course there's a really good way. That's why despite all that,
please tell us.
And yeah, it's just, it's like this job, if you really like it, it's like you're a kid that you're allowed to play with and answer questions that that they just make you wonder, and that's just absolutely amazing. You prove theories, you tested it. Having said that, there's always not bad, but the thing you need to come across is that like 95% of the time you're always wrong, and that's also can be hard sometimes. You work with failure pretty much, but upon failure then you build a lot of new insights and knowledge, and to know that you did that no one else in the world is doing or knows what you knew when you saw the microscope. That feeling is just priceless. It's worth the contract.
Well, thank you very much. Thank you very much. Thank you very much for your time. Unfortunately, we need to finish the event. I really appreciate your time and your effort, not only on this talk, but also on your passion for science and on trying to make it happen. And yeah, for sure, there are things that needs to be improved to change the scientific system, and because we all are going to benefit from that. Well, thank you very much, everyone, for your attendance, and thank you very much to the SRAP. Thank you very much to Instituto Cervantes for allowing this event to be possible. So thank you very much, and have a great afternoon. Bye. Bye.
Thank you.
Thank you, Raquel and Miriam. Thank you.